The overall goal of this protocol is to analyze dendritic routing of adult Drosophila medulla neurons in columns and layers, to characterize dendritic patterns, determine cell types based on morphology, and identify mutants.
This method can help answer key questions in the field of neuroscience, including mapping the brain circuitry and understanding the wiring mechanism of the dendrite.
The main advantage of this technique is that it parses out the dendritic properties in three-dimensional space, thus improving the understanding of dendritic wiring logic and functions.
The goal of this procedure is to acquire two image stacks of the neurons of interest in two orthogonal, horizontal, and frontal orientations. Using standard techniques, stain dissected fly brains with rabbit anti-GFP and an antibody that labels photoreceptor axons primary antibodies. Then label the primary antibodies with fluorescent secondary antibodies and clear the brain in 70% glycerol and 1x PBS. To mount the brain in the horizontal orientation, transfer a glycerol-cleared brain into a 20 microliter drop of anti-fade mounting medium deposited centrally on the microscope slide. Then on a cover slip, attach small patches of clay at the four corners to prevent the cover slip from crushing the brain. Now, under a dissecting microscope, position the brains in the ventral up position. Use the convex dorsal surface of the brain as a landmark to identify the orientation of the brain sample. This will provide a horizontal view of the neuron. Then attach the cover slip. Using a